Hey everyone, Professor Davis here, and we are going to dive into microbial metabolism. Now, metabolism was discussed a little bit when we were looking at the chemical properties video, and we introduced the idea of catabolism. Catabolism is the degradative, meaning it's breaking it apart, and the exergonic reactions. And remember that some of these are hydrolytic, meaning water goes in and it breaks the bonds. And so if you look here in this example, you can see that we have a polymer and we're going to add water. Energy gets released, and now I have a monomer plus a monomer. And this is where the water then goes. We then see here that we have the anabolism. Anabolism is the biosynthetic type of reaction, meaning we're building something. It's also endergonic, which means energy has to go into the building process. Some of these are known as dehydration because you're going to remove water out. So water is a product or an output. So here I have a monomer plus a monomer. Energy is going to go in. This creates the polymer and water is created here. Okay, so we have catabolism, we have anabolism. So here's an example here where I have a monosaccharide plus a monosaccharide, and we're going to form a disaccharide. This then results with water coming out of it. So is this a dehydration or a hydrolysis reaction? Well, since water is coming out as a product, it is dehydration. We then have to look and see, is it anabolic or catabolic? Am I building something or am I breaking something? Well, in this case, I'm building it into a disaccharide, so it is an anabolic reaction. Now, anytime we build anything, energy is required. Since energy is required, is it an exergonic or an endergonic reaction? This is an endergonic reaction. Energy has to go in. So when we're talking about the energy source that cells are going to use, we are really talking about ATP. And ATP works with a lot of these reactions by coupling with them, meaning pairing up. And so if you look here, you can see on the outside, it's showing you how anabolism and catabolism takes place, and it makes this kind of cycle. We build things, we tear them down. We build them, we tear them down. But in order for this to be successful, ATP is in the middle coupled with this, where the ATP energy fuels the anabolic reactions, but then it is going to be released by the catabolic reactions. Now, one thing to note though is these energy transformations are not 100% efficient. If you'll notice, we have arrows coming off the sides where heat is released. Some of this energy gets converted as heat. That energy is not usable by the organism anymore, okay? And so this just gives you some examples of those coupling reactions. So a concept here that you can see from the page before is that catabolic reactions drive anabolic reactions. So this AB being broken down into A plus B, energy gets released. That energy is used for C plus D to create and build a new CD. So the catabolic reactions are going to be fueling with their ATP the anabolic reactions. So this brings us to looking at metabolic pathways. These are the chemical reactions, and some of these chemical reactions are going to require enzymes to be present. Now, enzymes are really important because they can actually speed up chemical reactions and make them faster. Okay, it takes many different enzymes to catalyze all the reactions within cells because enzymes are specific. Okay, now since there's a lot of variety, we don't really need many of them though. Okay, cells don't have to make large quantities of each enzyme, they just need a lot of different types of enzymes. So let's take a closer look at the enzymes. So enzymes are organic protein catalysts. Okay, so they are proteins. We learned about proteins back in the chemical concepts chapter. We do see that when we see that they catalyze, they're a catalyst, they make things happen faster. So they speed up the chemical reactions. Now they do this by lowering the amount of energy required for the reaction to take place. So if we take a look at this particular picture, the red line is showing you what the regular reaction looks like. So we start here, we have to add energy to it, and then we can actually do the reaction. Okay, if you look in the blue, however, the blue is with the enzyme. And so it takes still some energy, but it's a lot less. It takes less, and it allows this to take place again faster and quicker. So the concept here that we need to make sure that we learn is that enzymes lower the energy of activation, and we can see that here in this chart. Now, enzymes are specific, okay? They are specific, and this is because if you remember, proteins have a very specific three-dimensional shape. When they make that three-dimensional shape, they determine the shape of their active site. Their active site is where the substrate or the reactant binds, and it can only bind to one thing and actually continue to still work. So because of this, they have what we call high specificity. They only work on one reactant. Now, enzymes normally get named by using ACE at the end. Not always, but for most of them they do. And so if you look here, if the substrate is urea, that's going to break down urea, the enzyme would be urase. Okay? If it's going to break down lactose, the enzyme would be lactase. So that's just an example of some of the enzymes and how they're named and the fact that they are very specific. So here is a chart that shows you some of the chemical reactions that are catalyzed within a different types of organisms. And so you can see that the enzymes are listed here and they all end in ACE with their class. It also tells you some examples of them. So like the cytochrome oxidase is circled here, as well as kinases, phosphate group. These are going to be really important because these are going to be enzymes that are important for ATP transfer, but also these are some of the enzymes that we can actually detect with certain kinds of biochemical tests. And so if a cell actually has the ability to use the cytochrome oxidase, we could do a test called oxidase to find out if it has that particular enzyme. So these are just some examples. Now, functionally, we do see that some of these enzymes may require the presence of some help, okay? In order for them to actually do their job and do it well and efficiently, they may need some help. And some of the help are going to be things like cofactors. So cofactors are inorganic metal ions, okay? And so they are inorganic. These would be metals like iron, nickel, things like that, okay? And what they would do is they would bind to the enzyme and then activate it. Then the enzyme is activated to do its job. Coenzymes are another thing that could be present. Coenzymes are organic molecules. These are going to be things like NAD+, FAD++, and CoA. And we are going to see these a lot when we look at the metabolism kind of pathway for microorganisms and even for ourselves, what our mitochondria would do. These a lot of times are synthesized by vitamins. So this is why it's important that you have certain vitamins present and certain kinds of nutrients present in the agar when you're wanting these different microorganisms to grow. Okay, so this is just an example. So some that are really important when we see where you get those particular enzymes from. If you look here, B2, vitamin B2 called riboflavin, it's used to help create FAD, which is flavin adenine dinucleotide. We also see that niacin is important, and this is going to be the active ingredient in NAD, which is an electron transfer as well. And so these are just showing you how some of the vitamins are very important in order for these different enzymes to function. So let's take a look at the function of these enzymes. So an enzyme is going to have their active site. Once the active site is open, the substrate can enter into it and bind. Once the substrate binds, we call this the enzyme substrate complex. That's with number 2. Once this complex is formed, we're going to see that the enzyme can change shape, and it will either build it or break it depending on what the enzyme's job is. In this particular case, we're starting with this green molecule, and we're going to break it into a blue and the yellow molecule. And so this substrate is converted into its products, and then the products are released from the enzyme. Now, if you'll notice, the enzyme didn't change. So the enzyme can keep working, okay? It can find more substrate, and it can continue its job, okay? So if we're looking at this one, since we're breaking it apart, is this an anabolic reaction or a catabolic reaction? Well, since it's breaking, the products are breaking apart, we see that it is catabolic. Now, enzymes are going to be influenced by a number of outside factors. These factors include things like temperature. Okay, so temperature can actually affect how well an enzyme works. We see that enzyme activity will have a optimal temperature, so it'll have a hard time working too low or too high of a temperature, and you can see that here in this picture. We also see pH. It'll have an optimal pH. Now this pH is going to be different for each type of enzyme. Some enzymes work better with acidic pHs like the ones in your stomach. Others work better in a more neutral pH. It just depends on the enzyme. And you can see it here in the picture where it creates a curve as well. Now you'll notice that both of these have little asterisks next to them, and that is because of the reason these guys don't work well at high temperatures or high pHs, or even low pHs, is because they denature, they unfold. And since they unfold, they're no longer usable. Remember, enzymes have to have a particular 3D shape. So if we look here too, we also see some other factors are substrate concentration. Okay, if there's a lot of substrate present, enzymes are going to work, their rate will be faster. Enzyme concentration is another one. If you only have a few of them working, it's not going to be as fast as if you have more. And then also the presence of something called inhibitors. Inhibitors are things that are going to stop the enzyme from doing its job. Okay, so if you look here with the heat, you can see that heat increases the collisions between the enzyme and the substrate, but enzymes denature if it gets too hot. We see the same thing in the pH. If the pH is too low or too high, we see that the enzyme will denature. All right, so let's look at some of this idea of inhibitors, okay, if an inhibitor is present. So when we look at this, enzymes have their active site, and they also may have another area where something can bind, which is called the allosteric site. Now with these, inhibitors are going to prevent the product from being formed, and they're going to do this by changing the shape of the enzyme. So competitive inhibitors are going to compete for the substrate's active site. They're going to take the spot. So this would be like if you're looking for a parking space, okay, and you go and some other little car or a motorcycle is in the space. It looks at first like you could pull into it, but then something is in the way, okay? So it's taking up that particular space, right? So it's competing for the space, or even a cart is in the way, right? That's a competitive inhibitor. A non-competitive inhibitor is going to be where it connects to an allosteric site. It's not actually taking up the active site, but because it binds, it changes the shape of the enzyme, and now the active site can't be used. So this, going back to my analogy with the parking lot, this would be like if you were trying to park in a parking space and two big pickup trucks are parked in each side, but they're right on the line. Okay, they're right on the line both sides. Well, that changed the shape of the parking space just enough that you may not feel comfortable parking there. All right, and so it changed that shape. Even though they weren't actually parking or in the spot you wanted, that spot's no longer usable, okay, because it changed the shape just enough to where you couldn't fit. Okay, so that's what we consider a non-competitive inhibitor. Now, these inhibitions, this idea of using inhibitors, can be used in what we call a feedback. This is a feedback inhibition. This is a normal mechanism that lots of cells use, and they do this to control the amount of product being produced. So if they have plenty of the product, they want to shut down the enzyme so that they're not making more when it's not needed. Okay, so let's look at how this works. So if my substrate is A, okay, and I want to eventually change it to something else, I'm going to have my enzyme. ACE is going to change it to B. B is then going to be changed to C with the enzyme BACE. And I'm only using this as an example where ACE is at the end to show you that it's an enzyme. C is going to be made into D with DACE. And then eventually we're going to finally get to E with the DACE enzyme. This E is going to be the product, okay? So in this particular case, it took 4 different types of enzymes in order to get to the product we want, which was E. Okay, now what then can happen is this final product can then attach to that first enzyme AACE. And when it attaches, it's going to temporarily inhibit it. It's going to attach to an allosteric site like you see here, and this makes it where A can no longer attach. Okay, so A can't attach anymore. Now this will continue to happen until we run out of E. Once the products get lower and we no longer have the E product, E will unattach from this enzyme and then the whole cascade can start again. All right, and so this is kind of like a light switch. It can turn on the pathway or it can turn off the pathway. Okay, so note that each of these steps is going to require its own enzyme. This is a type of negative inhibition, meaning we can reverse the condition. And if we have too much product, the pathway shuts off. If we have not enough product, the pathway turns on. Okay, so this is where we're not wasting energy, because if you have enough product, you don't need it on. Okay, just like if you have enough sunlight coming into your window, you don't have necessarily your light on. Okay, and so, but when it gets dark, you may have to turn the light on. Okay, same thing here. If we run out of the product, we'll have to turn the enzymes back on. And so this is what it kind of looks like more with an actual example. You can see that this substrate comes in and it's going to use intermediate A, intermediate B until you finally get the end product. So you have enzyme 1, 2, and 3. By the time you get to the end product is going to bind to enzyme 1. It's going to shut it off and it shuts the pathway down. Now remember, it is reversible. And when the final product does accumulate, it begins to bind and inactivate the enzyme. This shuts the pathway down. Okay, shuts it off. The pathway gets turned off. Now, if we run out of this end product, we can take it off of the enzyme A and it starts the pathway back up again. Okay, so this just gives you an example. All right, so this finishes up part 1 of microbial metabolism, where we focus mostly on the enzymes. We are going to see in part 2 that we're going to get into how does energy get produced, and we're going to look at those pathways like glycolysis, Krebs cycle, and even the electron transport chain. But that's going to be in part 2. So if you have any questions, please let me know.